embark on using these tools like genome editing tools to do functional genomics to try and characterize different regions of the genome is we want a way to sort through all those 3 billion letters in the human genome, all those A's, T's, C's, and G's, and efficiently pinpoint which regions of the genome might be involved in processes like cancer, which we just heard about in the previous talk. But it could be, of course, any disease or any phenotype. Just to focus on cancer for a second, though, this is kind of an urgent need because over the last 2 decades, we've been able to sequence more and more people, more and more, say, tumor samples. And we now are faced with finding thousands of different disease-associated variants, cancer-associated variants. And we know that probably not all those variants that we find are actually drivers of something like tumor proliferation or drug resistance or metastasis. And so the question becomes, as we sequence more and more people, more and more patient samples, how do we really distinguish between those mutations that are just along for the ride and the ones that are really driving some sort of disease-relevant phenotype? And can we somehow sort through those thousands of mutations in some kind of scalable fashion instead of looking at them really just one at a time? So that's one of the questions we'll try and address today, is how can we rapidly understand the effects, the functional effects of all these different variants? And as I said at the very beginning, the tool that we've been taking advantage of are these new programmable nucleases, many of them borrowed from bacteria that use them to fend off their natural predators. And it's, of course, hard to overstate the fact that this has really been a tool that's swept through biomedical science and is used in so many labs today, including our own. And I think one large reason that is the case is because they are so programmable. It's pretty easy to get these up and running and targeted anywhere in the genome. And I'll go over that in a little bit more detail in a few slides. But just to give you a roadmap of where we're going today, I thought I'd start with one recent story about looking for functional elements not in genes, but in that other large portion of the genome that's non-coding, that we think harbors many regulatory elements that can control genes. And there I'll show you a very specific example from drug resistance in melanoma, drug resistance that evolves to a very molecularly targeted therapy in melanoma. So that's going to be the first part of the talk. In the second part of the talk, I'm going to tell you about something that we've been recently doing. Instead of just focusing on really one cell type, or as we've done in the past, doing entirely kind of in vivo approaches where we have really too many cell types to pinpoint specific cell interactions, to actually look at genome editing in one cell and see how it reacts or communicates with another cell. And that'll be between a tumor cell and an engineered T cell, where we can look and ask questions about why new kinds of immunotherapies don't work in all patients who receive them. So just to dive into the non-coding genome, why should we care about this? So most of the genome, as you probably know, is non-coding. A little bit more than 98% of the genome does not code for proteins. And in 2012, the ENCODE group, which is consists of many of the same folks involved in the Human Genome Project. This is what they did in the following 10 years. They came out with this kind of landmark series of papers all published together in the same issue of Nature. And they said about 80% of the non-coding genome has some specific biochemical activity. That was the number they cited, which is actually a pretty large fraction of the genome as being functional. But estimates of around the same time and more recently have tried to look at function in terms of conservation of closely related species. The idea if something is truly functional, it should be conserved, right? It shouldn't be mutated away. And those estimates have been almost an order of magnitude lower, something like 8%, 10% of the genome. And so there's a little bit of a discrepancy here. Which one is it? Is it that 80% of the non-coding genome is functional, or is it something more like 8 or 10%? Kind of an additional motivation for why this is an important area to look at is that in genome-wide association studies, GWAS studies with common diseases like diabetes, heart disease, most of the genome-wide signal for drivers of these diseases have actually been found in non-coding regions, not in regions that code for genes, which are more of the rare variants. Those are not the common variants. So if we want to understand a lot of this common variation, we should make some efforts to understand the non-coding genome. Now, this is not to say that this is something that we can now do with CRISPR or with genome editing that wasn't possible before. There's many very established assays for non-coding function, things that look at physical interactions of the chromosome, the 3D conformation using things like chromosome conformation capture or other C assays, ChIP-seq to look at where transcription factors bind in the genome, looking at open and closed chromatin states. I mentioned conservation already. And also not just the primary sequence of the non-coding genome, but also other marks that we think are relevant for function, things like methylation of DNA bases or modifications of the histone proteins that the DNA is wrapped around. So we already have all these assays for function. Why do we need something else? So what I'm going to argue for is the reason that we need something else is that these are kind of indirect assays of function. And what we really would like to do, if we think that this particular region of the non-coding genome in this kind of 3D conformation is influencing a target gene that then has some influence on a cellular or organismal phenotype, is we'd like to go in here and either alter or mutate the sequence of this functional element and actually see the loss of this function or gain of some new function and somehow changing the phenotype of the cell. That's the idea with being able to target a specific editor to a specific region in the genome. And the editor that we're going to use in this context is something that I'm sure many of you are familiar with, which is this enzyme Cas9. We've already heard some nice talks earlier in the conference about this. And just so we're all on the same page, here Cas9 is schematized, the protein, as this yellow blob that's directed to a specific target in the genome by a single-stranded RNA, this single guide RNA, and I've drawn it here with the 5' side over here, 3' end over here. And the important thing to note is the bases here in red are a conserved scaffold that Cas9 uses to recognize its guide. And the bases here, the 20 bases on the 5' end here, Cas9 uses to find a target within the genome, and then as a nuclease, it can cut once it finds the site. And if we target it, of course, to an exon, this cut can result in a small indel mutation that can result in loss of function of the gene. If we target it to some intergenic region here, some non-coding region, this little indel can result in a mutation, which is the kind of thing that we might want to create to test function of non-coding regions. And one thing that we and now many others have done is take advantage of DNA synthesis technologies, similar technology that's used to make microarrays in the context of sequencing, but we just cleave these oligos off the array. And so here, instead of designing one guide RNA to direct Cas9 to just one region in the genome, we can design really tens of thousands, up to millions of these guides at a single time, just click print, and then have this library delivered to us really in one tube. And what we did now a couple of years ago to give you the context for entering into this non-coding exploration is we were interested in looking genome-wide, initially at protein-coding genes, to find drivers of resistance to a drug called vemurafenib. So just a little background on vemurafenib. So mutations in the BRAF gene, specifically this gain-of-function mutation, are found in up to 70% of melanomas. It's by far the most common driver mutation in melanoma, skin cancer, which is also a very common, very mutagenic cancer. And this kind of now famous example from data from Levi Garraway's lab I'm showing here of drug resistance. So in 2011, the FDA approved vemurafenib. It's a so-called kind of next-generation chemotherapy. It targets specifically mutant BRAF because regular BRAF is expressed in virtually all of our cells. And you can see a patient who carries this BRAF mutation that has several tumors here in this picture. You can see after treatment with vemurafenib, you see this initially this really quite amazing remission. Unfortunately, in almost all cases of patients treated with vemurafenib with extended treatment, you can see recurrence of these tumors. It comes back. And so we had initially wanted to ask what are all genes in the genome that might drive resistance to vemurafenib. And because this is kind of older work and is now published, this work I did with Ophir a few years ago, here what we initially did is we created this guide library that I just talked about to go to all protein coding genes in the genome to create loss of function mutations. And what we found, just to very briefly summarize, is we found a few genes where with multiple different guide RNAs different sequences that attack these genes, they were all enriched after treatment with vemurafenib, meaning that vemurafenib was unable to kill cells that had been modified with knockout mutations in those genes. And in this way, we were able to kind of both rediscover the two known mechanisms of vemurafenib resistance and then also highlight several other genes, including a few that I'm going to talk about now in the non-coding context today. So based on this, we took some of these top hits, some of these genes that we know when you lose their function in vemurafenib, when you lose their function and treat the cells with vemurafenib, they become resistant. Instead of targeting the genes themselves, what we did is we created CRISPR libraries to basically mutate every little piece of the non-coding genome, 5' and 3' of these genes. So we looked about 100 kb 5', so this is the transcription start site here, 100 kb 5', and 100 kb 3' of these genes. And we basically didn't do anything very intelligent to design these libraries. We basically selected every single piece of the genome that we could target with the Cas9 enzyme. In the end, we had about 18,000 guide RNAs targeting up to about 1 megabase of the non-coding genome. And I'm just going to focus on one of these right here, this gene called 3. Oh, yeah, so what is, I've been hinting at this, I didn't really tell you what the nature of the pooled screen is, and this is work that I did with Jason, who's now at Homology Medicines. What we do is we take these BRAF mutant cells, these are human melanoma cells, and it's important to emphasize that they're human because typically we don't think about human cells as a genetic model organism. We think about using Drosophila or mice or bacteria. So that's really a change in the CRISPR era, is now humans are a genetic model, or really anything is a genetic model organism. So each of these cells receives a different one of these non-coding manipulations, a different one of those little blue lines from the previous slide. And then we treat these cells either with the vemurafenib drug or with a control treatment, and we want to see which of these particular genetic manipulations is enriched after being treated with vemurafenib. That's the goal. And so here, what I'm showing you here is after 14 days of vemurafenib treatment, you can see that most of these guides really do nothing, as maybe we'd expect in these non-coding regions, but there's some small percentage, about 7%, that are really significantly enriched above this control treatment. And if we look at these in a genome browser style view, you can instantly start to make out features of this gene. So right here, with this gene called 3, you can see right over the promoters, this is a few kb before the transcription start site, you can see this nice peak right here. And what this is directly measuring is enrichment in vemurafenib. It's saying when we mutate the genome in this region, how well does this cell proliferate, this human cell proliferate in the conditions of vemurafenib? And as I mentioned, these cells all carry that BRAF mutation. Normally, they're going to be killed by this drug within this time scale. And so you can also see that within this first intron, there's some enrichment right here. We know there's many regulatory elements that tend to be within the initial introns. But really, throughout the 100 kb, 5' and 3', you can see there are distinct regions that have functional enrichment. So what we did was something kind of similar to that one slide that I flashed up about the genome-wide coding screen. There we looked for different guide RNA sequences that target the same gene that were enriched in vemurafenib. Here what we did is, since we don't have the idea of different genes, this library is designed to one gene, we looked for different guide RNAs that target in kind of nearby each other within the same spatial window that were simultaneously enriched. So these two guide RNAs here both seem to target within 40 bases of each other. And here, using ENCODE data, we can actually see a ChIP-seq peak for a transcription factor, YY1. In fact, this guide 2 lies directly over a consensus motif for the YY1 transcription factor. And what you can do is, if you just take this single guide, you put it into these cells, you can deep sequence and read out the mutations that are made by it. And you can see that several of these mutations are predicted to disrupt YY1 binding. But that's just a bioinformatic prediction. what actually happens in these cells. So we can go ahead and introduce these two guide RNAs and actually look by ChIP-qPCR, we can pull down the YY1 transcription factor and see, is it binding as well to these regions of the genome? And you can see in both cases, and especially this one that sits right on top of the YY1 peak, that binding of YY1 is significantly decreased after we edit at this site. And just as you'd expect, the expression of this nearby gene of CUL3 is also decreased. So it suggests that by disrupting YY1 binding, what we're actually able to do is kind of phenocopy loss of function of this gene, triggering the resistance of vemurafenib. So this is a way to actually look at a specific non-coding mutation and say, here's how it might have something to do with this relevant phenotype. So we did this in a few other regions where we found multiple guide RNAs that were enriched together and where we had a good antibody where we could pull down a specific transcription factor. But at the end of the day, this is kind of a really unsatisfying assay. Here you've taken this super high throughput, whatever, 18,000 guide RNA screen, and now you're validating it one by one with a super painful kind of looking for transcription factors, testing antibodies. So we thought, is there a way we can do this in a little bit more high throughput fashion? And for this, we turned to look at globally transcription factor binding by looking at open chromatin regions, which can be done with traditionally one of these two assays, DNase hypersensitivity sequencing or ATAC-seq. And here we use this one, ATAC-seq, which basically measures openness of chromatin or lack of nucleosome occupancy by the ability of a transposase, a bacterial transposase, to insert in these open chromatin regions. The idea being that if it's open, it's easier for the transposase to insert. And so we did this not only in this human melanoma cell line that we had used, but we did it in a few other different human cancer-derived cell lines, a breast cancer and a glioblastoma. And you can see our data kind of lines up pretty well with the ENCODE DNase open chromatin data. And what we found, this is just kind of one example, but what we found is that the enriched guides from our screen tended to co-localize with these open chromatin regions. And so if you look across the entire 200 kb region that we screened, you can see an enrichment for these functional elements as detected by the CRISPR screen with specifically open chromatin regions in melanoma, but not in breast cancer or glioblastoma. And it's true whether you use our data or the ENCODE data. So that's nice to see it line up with that. What about other well-known hallmarks of non-coding function, like these epigenetic marks that are associated with enhancers or promoters? So here what we did is we examined some of these marks like H3K4 trimethylation, which is often found at promoter regions, and some of the marks like H3K27 acetylation that are found at enhancers or other distal regulatory elements. And what we found when we took 10 of our enriched guides within 1 kb of the transcription start site of the gene, we found that after mutation at these regions, you actually see a decrease in this promoter-associated functional mark. And the same thing for one of these more distal marks, which is H3K27 acetylation, when we took 10 of these, individually took 10 of these guides at distal regulatory elements. A little bit, there was also a shift with this H3K4 dimethyl, but it was a little bit less significant. Another hallmark of function in the non-coding genome, as I mentioned, is this idea of the conformation of the chromosome itself, that it's not that the genome is kind of randomly bunched together, but it tends to be wrapped and folded in a pretty reliable, consistent way. And we wanted to understand what was the interaction between how the genome was folded and these functional elements that we detected in this non-coding CRISPR screen. And so here I'm just showing you a chromosome conformation capture assay. So basically what these peaks here mean is how, in 3D space, how close are these different regions, either 5' or 3' of the gene, how close are they to our bait region, which was selected to be the promoter of CUL3. And if you imagine just kind of water filling up this graph on the y-axis, so that's increasing the 3C interaction frequency, you can see there's a real clear monotonic relationship between 3C interaction, that is, how close a region of the genome is to the promoter, and how enriched it is in the CRISPR screen. And what this tells us is that the functional elements in 3D space tend to be actually quite close to the promoter of the gene that they're controlling, even if along the two-dimensional chromosome, they're quite far away. And so the argument that I kind of want to put forward here, even though there's, I think, a lot of work left to be done, is even though we have all these other hallmarks of non-coding function, whether it's these epigenetic marks or open and closed chromatin, transcription factor binding, or kind of 3D interaction, that these functional screens, these CRISPR screens, which I showed you just one of here, add a different layer on top of all of this, because it really directly connects regions of the genome with disease-relevant phenotypes or traits. So with kind of that as the first part, I'm going to switch gears a little bit and tell you, kind of go a little bit perhaps backwards in time to a genome-wide protein coding screen, but something that we just did recently that I think is really exciting. And because of the time limitation, I'm going to skip over some other work, but I wanted to say as a way to introduce this, another thing that we did in addition to these kind of assays with single cell types in a dish is we wondered, could we do this in vivo? And so a few years ago, we actually did one of these genome-wide CRISPR screens in a mouse. And there it was really exciting because we could look at phenotypes like metastasis, which are very difficult to analyze in the context of in vitro cell culture. But in some ways, it was very unsatisfying because we didn't really know which cells might be driving some of the interactions with the modified cells that we had put into the mouse when they metastasized. And so here we had kind of wanted to bridge this gap between the totally in vitro world and the fully in vivo screens that are a little less controlled. And so this was an effort in that direction, and this is a collaboration with Nick Restifo's group at NCI. So as many of you probably know, that immunotherapy has been probably one of the biggest breakthroughs in the last decade in cancer treatment. It's really revolutionized cancer in places where normally patients were kind of not able to have any treatment in a late-stage cancer. But even though there's a subset of patients where immunotherapy works really well, and I should just say immunotherapy, of course, is harnessing the immune system to fight cancer, it's been observed for over 20 years that there are different mutations that seem to render certain patients resistant to immunotherapy. So this is work from Nick's group also 20 years ago with adoptive T-cell therapy, noticing that mutations in beta-2 microglobulin render these patients resistant to adoptive T-cell therapies. And this is more recent work with PD-1 inhibitors, which is one of the more very popular FDA-approved therapies, that mutations in a few different genes also, including B2M, result in resistance to this immunotherapy. So the question we wanted to ask is, can we, using these cool CRISPR library tools, can we predict genome-wide what are all mutations that drive resistance to cancer immunotherapy? And the idea here is to modify these tumor cells, to knock out every single gene in the human genome, and then challenge them with T cells that are normally engineered to go after that cell and kill it. That's the basic idea. And so what we did here is just show you that the system works is we have these specific T cells. These are donor T cells, primary CD8 positive T cells that we modified with the NY-ESO TCR, which is a T cell receptor that recognizes a very specific antigen. And you can see it's only able to kill the cells in the presence of the antigen. Cells that don't display the antigen just are unrecognized by these T cells. And we took this very famous example from the literature, beta-2 microglobulin, which is one of the two proteins that's involved in MHC class I presentation of antigens on the cell surface. And we went ahead with Cas9, we knocked out beta-2 microglobulin. And you can see when you knock out B2M in these human melanoma cells, basically they all survive the T cell onslaught. Versus with just a control treatment, the T cells are very effective and kill them within just a few hours. Okay, so this system seems quite specific, seems to work in the way we think with antigen presentation. So here, we basically, around the same time, we designed some new genome-wide CRISPR libraries that target virtually every gene in the genome and almost every microRNA and miR base. And clone them into a lentiviral vector, which is what we use to deliver this. I just say one thing, since I'm talking about genome-wide libraries, but a lot of the work that we're doing in my lab now is actually trying to look at very focused sets of genes like transcription factors or chromatin modifiers. And just as a way to get the word out, if you're interested in doing these kinds of very custom library designs, we now have a tool available on our website designed by a very talented undergraduate student that really lets you, in seconds, design a custom CRISPR library to target virtually any subset of genes. And this uses kind of our most advanced design pipeline that both looks at on-target, off-target, and also takes into account the structure of proteins when designing guides. Okay, but back to our genome-wide library. So this looks slightly more complicated than the slide I showed you in the non-coding screen, but it's essentially the same assay. So here, we're in these cells, we're basically knocking out every gene in the genome instead of non-coding regions, mutagenizing genes. And instead of challenging them with a vemurafenib treatment, here what we're doing is the drug here, the drug, is engineered T cells engineered to kill these melanoma cells. But the readout is very much the same. We want to see what particular guide RNAs enable these cells to survive the T cell killing. And we were really happy to see that our top 2 hits, so this is out of 20,000 genes in the genome, hit number 1 and 2 in this CRISPR screen are actually the 2 components of antigen presentation, HLA and B2M. And amongst the top 0.1% of the library, which is the top 20 genes out of 20,000, you pretty much have all the major players in antigen processing and presentation. So this gives you an idea. I mean, none of this is novel biology. These are well implicated in antigen presentation and resistance to immunotherapy. But it gives you an idea of the robustness of the tool and the technology, which is nothing about our experimental abilities, but really how specific these CRISPR enzymes are. Okay, but this was known, so how do we discover something new? So here we looked a little further down the list than the top 20, and we took some of these genes. I'm not going to be able to get into the details, but we were interested in seeing, do the hits we find in this one human melanoma cell line with this one engineered T cell, do they generalize across different melanomas? What about different cancers? NY-ESO is actually an antigen found in other cancers too, and also other antigens than NY-ESO. And what about in the in vivo case? And I'm just going to show you one particular gene that we pursued a little bit, and that's this gene called the apelin receptor. And here what we did is we took a mouse melanoma cell line, we knocked out this apelin receptor, and then we transplanted these into mice and gave them adoptive T cell therapy in an antigen-matched way, similar to what we did with the human cells. And what you can see here is that with the knockout of APLNR, you can see that the tumors, though it's not quite as huge of a stop as knocking out all antigen presentation, it's quite a significant difference in tumor growth and also in survival of these mice after receiving immunotherapy, suggesting that this really is a bona fide block to immunotherapy. Another thing we were very interested in doing is, because these kinds of studies in mice are difficult